Welcome back, guys. Today we are going to discuss this important topic of cell organelles. So, cell organelles are the important components of a cell, how they functions are some of the importance of clinical importance. So, let's discuss them one by one. The first organelle we are going to discuss is endoplasmic reticulum. Endoplasmic reticulum. This endoplasmic reticulum is of two types. We all know what are the two types. First is the rough endoplasmic reticulum. This in short RER. Next is smooth endoplasmic reticulum, in short SER. So there are two types of endoplasmic reticulums, rough and smooth. So why this type of reticulum is known as rough? So let's see. This is the surface of reticulum, and these are the presence of granules. So presence of granules gives a rough visual to endoplasmic reticulum to form rough endoplasmic reticulum, right? The smooth endoplasmic reticulum is agranular. This rough endoplasmic reticulum has granules. So is rough endoplasmic reticulum, the smooth endoplasmic agranular. These granules are actually the ribosomes, which are required for protein synthesis. These granules are actually the ribosomes, which are required for protein synthesis. Let's discuss one by one. First is the rough endoplasmic reticulum. This rough endoplasmic reticulum, as we have seen, these granules are ribosomes. So there occurs synthesis of proteins, which is actually called translation. After the process of transcription or replication of the DNA, there occurs the synthesis of proteins in the ribosomes, process of translation. These proteins are required for proper functioning of cell. That is, there occurs folding of proteins by chaperones. Chaperones are also known as the heat shock proteins. This folding of proteins occurs by chaperones. Chaperones are heat shock proteins. This is a very frequent question asked in the exams. The chaperones are also known as heat shock proteins, or the heat shock proteins involved in folding of proteins are called chaperones. So we have seen this folding process of proteins is required for proper functioning. Any defect in this folding leads to formation of misfolded proteins. So the defect in this folding, any defect in this folding leads to formation of misfolded proteins. These misfolded proteins, when accumulated, is toxic and can lead to cell death. So one of the most common causes of cell death is accumulation of misfolded proteins. Cell death is an important topic which will be discussed in details in pathology in our second year. For now, we have to remember is most common cause for cell death is accumulation of misfolded proteins. To prevent this accumulation of misfolded proteins, the endoplasmic reticulum destroys the misfolded proteins. This process is called as endoplasmic reticulum associated degradation, ERAD, ERAD, endoplasmic reticulum associated degradation, which is the process of this endoplasmic reticulum destroying these misfolded proteins. So how this endoplasmic reticulum destroys, we are going to see in this ERAD process. Misfolded protein is combined with ubiquitin. This compound ubiquitin, misfolded proteins are combined with ubiquitin. This process is called tagging. Process is called tagging to form proteosomes. Proteosomes, which then is degraded. So in this endoplasmic reticulum associated degradation, these misfolded proteins which are toxic on accumulation is combined with ubiquitin. This process is called tagging, which are then converted to proteosomes and are then degraded. So from all this we have seen that the rough endoplasmic reticulum does the quality control of proteins. This is an important point. Rough endoplasmic reticulum is involved in the quality control of the proteins, as it destroys the misfolded proteins which can be toxic and can lead to cell death. So we are going to see some misfolded protein diseases. I'm going to see some of them. First one is Prions disease. Now we must all be familiar with this disease, the Prions disease, which occurs in the CNS. In CNS, normal protein, they are converted to PRPC proteins. CNS normal proteins are the PRPC proteins. These PRPC proteins gets misfolded during the formation or metabolism. These PRPC proteins gets misfolded, which are toxic. Misfolded PRPC are toxic. This leads to a very common CJD, which is Creutzfeldt Jacobs disease. You must be familiar with this disease, CJD, Creutzfeldt-Jacobs disease, which is due to misfolded toxic PRPC proteins in the CNS. Next, we are going to see another disease in the misfolded proteins is A beta amyloid protein. A beta amyloid protein. This accumulation causes Alzheimer's. A common Alzheimer's disease is due to accumulation of A beta amyloid proteins. This we need to remember. This is a clinically significant points. Prions disease and Alzheimer's. Prions disease is due to in the CNS misfolded PRPC proteins, and Alzheimer's is due to A beta amyloid proteins accumulation. So this is about rough endoplasmic reticulum. Second, we have seen rough endoplasmic reticulum. Second, we are going to see is smooth endoplasmic reticulum. It is smooth due to absence of granules, that is the ribosomes. Agranular. So agranular, no ribosomes. So some of the functions of smooth endoplasmic reticulum we are going to see. First is synthesis of steroid hormones. It's the form of lipids. Synthesis of the steroid hormones takes place in the smooth endoplasmic reticulum. Then we have got in muscles. This form of smooth endoplasmic reticulum is called SR, that is sarcoplasmic reticulum. Sarcoplasmic reticulum is the storage for calcium during muscle contraction. During muscle contraction, calcium is needed. So this sarcoplasmic reticulum is for storage of calcium during muscle contraction. The next, it is involved in drug metabolism. How? It causes detoxification in liver cells. We know this detoxification in the liver cells is caused by cytochrome P450 enzyme or cytochrome P450 group of enzymes. It's a very important point to be noted. Cytochrome P450 enzyme is often involved in detoxification of drugs which is present in the liver cells. This cytochrome P450 is associated with this smooth endoplasmic reticulum. So these are three functions of smooth endoplasmic reticulum: synthesis of the steroid hormones, in muscles the sarcoplasmic reticulum for storage of calcium during muscle contraction, and in the liver during the detoxification process the cytochrome P450 enzymes in the drug metabolism. So this is all about our endoplasmic reticulum. Next we are going to see the second organelle, which is Golgi apparatus. Golgi apparatus, in short GA. This Golgi apparatus is always closely associated with endoplasmic reticulum. The Golgi apparatus is always closely associated with endoplasmic reticulum. So these two organelles are called twin organelles. These two organelles are very closely associated with each other, so these two are called twin organelles, Golgi apparatus and the endoplasmic reticulum. So this Golgi apparatus, when close to rough endoplasmic reticulum, which is for protein synthesis, this Golgi apparatus, when closely related with rough endoplasmic reticulum involved in protein synthesis, the proteins go for Golgi apparatus modification. This protein synthesized goes for modification in Golgi apparatus. In other words, this protein synthesis is called translation, and this Golgi apparatus modification of proteins is called post transcriptional modification. This translation, after which in Golgi apparatus undergoes post-transcriptional modification. Proteins undergoes post-transcriptional modification in Golgi apparatus after synthesis in rough endoplasmic reticulum. So we are going to see one by one. First, the proteins when synthesized in the Golgi apparatus adds carbohydrate side chains. Golgi apparatus add carbohydrate side chains to the proteins to form glycoprotein. So carbohydrates combined with proteins are glycoproteins. This is to enhance the stability of proteins. This post-transcriptional modification is often done to stabilize the proteins. So the proteins, when added with carbohydrate side chains to form glycoproteins, it enhances the stability of the proteins. This process is commonly called glycosylation. Glycosylation. This process is called glycosylation. Next, proteins. The same Golgi apparatus, it adds mannose 6-phosphate. This is mannose 6-phosphate. The Golgi apparatus adds mannose 6-phosphate. This process is called tagging. The proteins get tagged with mannose 6-phosphate in Golgi apparatus. This tagging is important because after this it only goes to lysosomes. It only goes to lysosomes. This is called sorting of proteins. This is called sorting of proteins. Sorting of proteins, in which the tagging of proteins takes place with mannose 6-phosphate, and it only goes to lysosomes. And in lysosomes we know that destruction of molecules takes place. So in absence of mannose 6-phosphate, tagging, in absence of this mannose 6-phosphate tagging, there occurs eye cell disease. In this disease, the inclusion bodies accumulate, which can be further toxic. So this mannose 6-phosphate tagging leads to destruction of molecules in lysosomes, but in its absence there occurs eye cell disease, and it's the inclusion bodies accumulate, which can be toxic. So we have to see two forms of Golgi apparatus modification of proteins. We are going to see some parts of Golgi apparatus. There are 3 parts of Golgi apparatus. 3 important parts of Golgi apparatus, which are the cis end. Cis end is close to rough endoplasmic reticulum. Cis end is close to rough endoplasmic reticulum and is the receiving end. As this end is close to rough endoplasmic reticulum, the proteins formed enters the Golgi apparatus through this cis end. So this end is called receiving end. Second part is the Golgi apparatus proper. GA proper, Golgi apparatus proper. So this functions as performed. Functions are performed, that is post-transcriptional modification and sorting of proteins. This two process occurs in form of vesicles. In Golgi apparatus proper, it does the functions of post-transcriptional modification and sorting of proteins. It occurs in the form of vesicles. Third part is the trans end. Trans end, which is away from rough endoplasmic reticulum. Since the cis end is close to rough endoplasmic reticulum, this trans end is away from rough endoplasmic reticulum. This is the releasing end. The cis end is the receiving end, the trans end is the releasing end, in which after these two processes, these vesicles, release of vesicles takes place. The release of vesicles takes place in the trans end. The vesicles, after the processes of protein sorting and post-transcriptional modification, the vesicles which are formed are released from the Golgi apparatus from the trans end. Receiving end is the cis end, releasing end is the trans end. So this is all about the Golgi apparatus. It's very closely associated with endoplasmic reticulum. It is also called twin organelles. Third organelle we are going to see is lysosomes. The third organelle we are going to discuss is the lysosomes. Lysosomes, we all know, it is concerned with destruction. Lysosomes are concerned with destruction, so for which they are also called as the recycle bins of cells. The destruction of molecules takes place in the lysosomes, so these are called recycle bins of the cells. We're going to see some Facts about this lysosome. So this is a lysosome. This lysosome has an pump called H plus ATPase. It has a pump called ATPase, H plus ATPase, which is also called as proton pump. This proton pump helps in accumulation of these H plus ions inside accumulation of H plus ions through this proton pump. So the inside pH will fall as H plus are accumulated more and more inside the cell. So the inside pH will fall, which means it will be acidic pH. This is called acid mediated destruction. This is called acid mediated destruction. These lysosomes are also called suicidal bags or residual bodies. Suicidal bags or residual bodies. The enzymes in lysosomes which are concerned with destruction are acidic hydrolases and acidic proteases. So there are a lot of enzymes at the lysosomes, but we are going to discuss giving these two important examples, acidic hydrolases and acidic proteases. Why this word acidic? Because you have seen here, with the help of this proton pump, H plus accumulates, which converts the inside pH of the lysosome into acidic, called acid mediated destruction. So the enzymes associated lysosomes are named acidic hydrolases and acidic proteases. Hope you understand this. The functions of lysosomes is a process called autophagy or self destruction. Functions of lysosomes are autophagy or self destruction. So how this process occurs, we are going to see. During starvation, these lysosomes engulf a mitochondria. So during starvation, these lysosomes engulf a mitochondria. This engulfing of mitochondria leads to its destruction, which leads to release of proteins. The destruction of mitochondria leads to release of proteins. These proteins undergoes metabolism and releases energy. Energy is released by metabolism of proteins, which are released by destruction of mitochondria during starvation. During starvation, lysosomes helps in production of energy by this process. So We have seen how lysosomes function, why the inside of lysosome is acidic. This is important. This H plus ATP is a proton pump. It's called the recycle bins of cells, or suicidal bags, or residual bodies of cells. It is involved in acid mediated destruction. During starvation, lysosomes engulf the mitochondria, and its destruction releases proteins, which undergoes metabolism and releases energy. The next organelle we are going to see is peroxisomes. These peroxisomes are involved with hydrogen peroxide. Hydrogen peroxide, this name comes peroxisomes. This is hydrogen peroxide. Hydrogen peroxide is a free radical form. This hydrogen peroxide is a free radical form. This accumulation of free radicals leads to diseases. So this hydrogen peroxide accumulation, which is a free radical, can lead to diseases. So this peroxisome causes destruction of hydrogen peroxide. This destruction is done by an enzyme called catalase enzyme. This catalase enzyme is involved in the destruction of H2O2 by peroxisomes. So this enzyme is important to remember. Excuse me. Very important. Along with the cytochrome P450 we discussed above in the smooth endoplasmic reticulum. Then the second function is oxidation of fatty acids. This peroxisome is also involved in oxidation of fatty acids. Which fatty acids? Long chain and very long chain fatty acids. In lipid metabolism, in biochemistry modules, we will see how peroxisomes are involved in oxidation of long chain and very long chain fatty acids. These are long chain fatty acids are LCFAs, and very long chain are BLCFAs. These are the short forms, LCFAs and BLCFAs. So oxidation is done by the enzyme phytanic acid oxidase. Phytanic acid oxidase is involved in the oxidation of fatty acid. This enzyme absence can lead to this disease, Refsum's disease. Very important clinically found disease, Refsum's disease. Refsum disease can also be called as a peroxisomal storage disorder, as the phytanic acid oxidase, which is involved in oxidation of fatty acids, which is a component of this peroxisome. So this Refsum's disease, which occurs due to absence or deficiency of phytanic acid oxidase, is a peroxisomal storage disorder. This Refsum's disease is a peroxisomal storage disorder. It's an important concept. You need to remember this. So these are all about peroxisomes. Next we are going to see about important mitochondria. It is a very important cell organelle for discussion. So this mitochondria, as we all known as powerhouse of the cell. Why? Because it is associated with ATP synthesis. That's why it's called powerhouse of the cell. Mitochondria involved in ATP synthesis, so it's called the powerhouse of the cell. So we're going to discuss some functions of ATP synthesis. First we are going to see is oxidative phosphorylation. So after ATP synthesized, so oxidative phosphorylation takes place. The next is apoptosis process mediation. Oxidative phosphorylation and apoptosis process mediation. So apoptosis we have discussed in the cell membrane model. So programmed cell death due to inaccurately positioned lipids on the cell membrane surface. So these two are the functions of ATP synthesis of mitochondria. We're going to see the structure, very basic structure of mitochondria. And it's a very basic structure of mitochondria. This is the outer mitochondrial membrane. This is inner mitochondrial membrane. The mitochondria contains two membranes, the outer mitochondrial membrane and the inner mitochondrial membrane. This inner mitochondrial membrane, as we have seen in the cell membrane module, which is high protein content, about 70%. Maximum highest protein content is found somewhere in inner mitochondrial membrane, about 70%. This inner mitochondrial membrane contains convolutions. These convolutions, as we see here, these convolutions, these are called cristae. These are called cristae. This cristae contains enzymes of oxidative phosphorylation. You see here and above, these functions of ATP synthesis for oxidative phosphorylation. So the enzymes for this oxidative phosphorylation lies in the inner mitochondrial membrane, and this convolution is called cristae. Cristae are present in the inner mitochondrial membrane. In between these two membranes, this space is called intermembrane space. Intermembrane space, the space between two membranes of mitochondria. Very simple understanding. Now we are going to see mitochondrial DNA. We are going to discuss this mitochondrial DNA because this mitochondria has its own genome. This mitochondria is one of the organelles that has its own genome. This genome is double-stranded DNA with nearly 16,500 base pairs. dsDNA with 16,500 base pairs. This mitochondrial DNA. This mitochondrial DNA inheritance is usually maternal. This inheritance is usually maternal. That is, this inheritance is derived from exclusively ovum. So maternal inheritance derived exclusively from ovum. So we have seen this inheritance of mitochondrial DNA. Mitochondrial DNA susceptible to mutations. It is 10 times more in comparison to nuclear genome. It's 10 times more in comparison to nuclear genome. Is mutation in the mitochondrial DNA, and there is ineffective DNA repair. There is 10 times more susceptible to mutation and ineffective DNA repair. So these are some of the features of mitochondrial DNA. Mitochondria has its own genome. It's a double-stranded DNA. So the disease, I want to see some diseases. It affects the organ with high metabolic rate. Affects organ with high metabolic rate. Why? Because there is defect in energy production. So mitochondria is the energy powerhouse of the cell, involved in ATP synthesis, the energy currency of the cell. So there is defect in energy production. How some organs are affected, we are going to see. First are the skeletal muscle. Skeletal muscle, there is altered contraction and relaxation. There is altered contraction and relaxation, which is accompanied by lactic acidosis. In the skeletal muscle, there is altered contraction and relaxation, which is accompanied by lactic acidosis, which leads to fatigue. So fatigue in the skeletal muscles occur. Then there are gastrointestinal tract disturbances. The GIT tissues are disturbed. Then in the CNS, there is seizures, and person develop mental delays. In the CNS, there occur seizures and developmental delays. Then there is altered growth. There is altered growth, and finally they are highly susceptible to infections. These are some of the features. Disease which affects mitochondria. Skeletal muscle is affected. Disturbances in the gastrointestinal tracts. Seizures and developmental delays in the central nervous system. Altered growth. Person is highly susceptible to infections. So this is all about this mitochondria. Mitochondria involved in ATP synthesis for oxidative phosphorylation, and so it's also known as the powerhouse of the cell. Last important organelle we are going to discuss is nucleus. This is the important organelle to be discussed. It's present in all the cells that divide. It's present in all the cells that divide. It's in control of the cell functions. It's in control of the cell functions. Like our CNS is the controlling center, this nucleus is the controlling center of the cell. This nucleus contains chromosomes. Nucleus contains chromosomes which are the blueprint for expression. These chromosomes are the blueprint of expression, which are then the DNA, the deoxyribonucleic acid. Nucleus contains the chromosome which are the blueprint for expression, or the DNA. So let's see some of the features of the DNA. So this is a DNA molecule. This DNA contains histones, proteins. Histones are the proteins. This DNA combined with the histones forms the chromatin. This chromatin is formed by a combination of the DNA and the histones. This chromatin contains a repeating structural unit called nucleosome. This chromatin contains a repeating structural unit called nucleosome. Further details we will learn in the biochemistry modules of nuclear biochemistry. For now, remember this nucleosome is the repeating structural unit of chromatin. Then in the nucleus we have another structure called nucleoli. This nucleoli contains granules. These granules are RNA and ribosomes. This nuclei contains granules which are RNA and ribosomes. This, as we know, gives rise to proteins. RNA and ribosomes are involved in the translation and gives rise to proteins. So this part of DNA, they contains genes. This part of DNA contains genes which are the units of heredity. Units of heredity are genes which are the part of DNA. Not the entire DNA is involved in the hereditary. Part of DNA contains the genes. which are the units of hereditary. So we are going to see a basic structure of nucleus. This is the nucleus. This is one membrane and this is another membrane. This is the outer nuclear membrane. Very simple. This is the inner nuclear membrane. Simple to understand, nothing fancy. Outer nuclear membrane and inner nuclear membrane. Along this membrane, we are going to see that transportation of substances occurs both inside and outside. This transportation occurs through NPC, that is nuclear pore complex, that is for to and fro movements of substances. So this NPC, nuclear pore complex, for to and fro movements of substances. The proteins involved here are exportins. Simple to understand, to outside transport. For outside transport, exportins, and there are importins, that is to inside transport. For exportin for outside transport and importance for outside transport. These two are present in the nuclear pore complex for to and fro movements of the substances. So this is all about nucleus, and here we conclude our discussion on the cell organelles. A simple topic, nothing complex to understand. We all know majority of the things from our class 11th and 12th. Just a little bit addition of some important clinical topics like these misfolded proteins, which are important pathologically, this Prions disease and Alzheimer's, and how this Golgi apparatus is involved in tagging, how its disorder leads to inclusion bodies disease, eye cell disease. Then there are lysosomes, which are involved in autophagy, important to remember. Then we have discussed peroxisomes, removes destruction of free radicals with the help of catalase enzyme, and also involved in oxidation of fatty acids with the help of phytanic acid oxidase. The absence of this leads to Refsum's disease. So peroxisome storage disorder. Then we discussed mitochondria, how it has its own genome, the mitochondrial DNA, and its some features. The disease of mitochondria, how the body is affected. Then we discussed nucleus, the control center of the cell, and membranes of the nucleus. We discussed nucleosomes, DNA, histones, and nuclear pore complex. So that's all, guys. Make sure to revise the topic. Thanks for watching. I will catch you in the next one. Good luck.